immunology, and specifically today we're talking about applications of immunology. And what that means is basically there's very unique properties about immunological systems, specifically antibodies, that make them very useful tools for use in research, in biotechnology, in therapy, in clinical diagnostics. So we're going to talk about today about how we use those principles from immunology to benefit us in other ways. So the main use, clinical use of immunology principles is in the use of vaccines. Okay, we've talked about vaccines to some extent already. We'll talk about them in more detail right now. So we've talked about this in the very beginning of the semester, where the very first vaccine was really variolation, which was taking the actual disease-causing agent and inoculating it into the skin, and then seeing if those people that got inoculated with the smallpox were then resistant to smallpox infection. And it turned out that they were. Okay, that turned into what's more modern day vaccination, which is taking some form of the microbe, not the actual living microbe itself that caused the disease, but something very similar to it or some form of it, and using that to inoculate, and then you get the benefits of being immune to that disease in the future. So here's just kind of an intro video.
One way to gain immunity to a particular disease is to actually contract the disease. However, with many diseases, this could lead to serious illness or even death. Vaccines are a method of inducing immunity without the person contracting the disease. A vaccine is a solution containing a weakened or killed pathogen or parts of the pathogen that's administered, usually through injection, to a healthy person. When a person is vaccinated, the parts of the pathogen that are antigenic will be recognized by the adaptive immune system. B cells with the matching B cell receptor are activated and undergo clonal expansion, ultimately producing antibodies and memory cells. With some viral vaccines, the virus actually replicates inside the host cells and triggers a cell-mediated response, activating cytotoxic and helper T cells and resulting in the production of memory T cells. Should the same pathogen enter the person months or years later, it would be recognized by the memory cells. The memory B cells would become activated rapidly, producing large amounts of antibody. The antibodies attach to the pathogen and initiate destruction of the pathogen by phagocytosis or complement. Should a viral pathogen enter the host cells, memory cytotoxic T cells would recognize that infection and initiate apoptosis of those host cells early in the infection stages. Thus, the person would not become sick.
So again, vaccination is just a way to kind of put the body on alert that if it comes in contact with these pathogens in the future, you already have these memory cells in your serum, and they can act much quicker rather than seeing it for the very first time, and they have to start a brand new, fresh immune response. So here's just an example of one of the more successful examples of vaccines from the last century or so. The measles vaccine. So on the y-axis, you can see these are the number of cases reported per year. So you can see about almost 500,000 people were being contracted with the measles in the mid-60s. The vaccine was licensed in, looks like, 1964. And you can see the very significant decrease in the number of people that are getting the measles over time. And you can see right now it's a very low basal level. And of those people that are getting the measles, it's those that don't get vaccinated against it. So I'm going to spend some time talking about the different types of vaccines. There's actually 5 different types. And I have examples of each type of current vaccines that are given. Most of these are given to children. But again, a lot of you guys probably have some familiarity with some of these vaccines. Either you have children, or you remember getting them as a child, or you just, because a lot of you guys are in the health profession, you hear about them. So there's the attenuated whole agent vaccine. So the example there would be MMR, measles, mumps, and rubella. So this is something that's given, to cover 3 different diseases. It's given to infants for the most part. Attenuated whole agent vaccine basically means you either take the virus or the bacteria, whatever it may be, and it's a weakened form of that microbe. So it can be weakened, it can either be mutated in a way that makes it weakened, it can be heat treated, it can be treated with different kinds of chemicals to make it weaker. Okay? What that does is it allows a pretty strong immune response because it is still living organisms, but it's weakened to the point where it can't give you an active infection. It's just strong enough to boost the immune system, but not strong enough to actually give you an active infection. Okay, the next subtype would be the inactivated whole agent vaccine. The screen changed color.
Yeah.
I'm not going to start messing with stuff. All right, so inactivated whole agent vaccines, again, they're whole microbes. So again, for a bacteria or virus, it's the whole bacteria or the virus, but it's completely inactivated, means it's basically dead. So a bacteria or virus that's been completely killed, but it's still in its original whole form. And polio vaccine is an example of that.
But if you put a dead virus in, what's, like, how do you...
So the dead virus, basically any antigens that are on the outside of the envelope of the virus will form an antibody immune response. But you're right, you won't get the cytotoxic T cell response because that requires active replicating virus inside of cells.
But it still works.
Depending on the virus, it still works to give you an antibody response, but you won't get a T cell response.
Okay.
So in this case, polio, it does work for that type of virus. Toxoid. So again, we've talked about cases where it's not the bacteria itself that causes the infection, it's a toxin that the bacteria produces. And so you can actually take that toxin, again, in a small dose or an inactivated form, and inject that directly, causing an antibody response against the toxin itself, not against the bacteria, but against the toxin. And then you'd be protected against that toxin. You wouldn't be protected against infection from the bacteria, but you'd be protected against the toxin. So that's a toxoid vaccine. There's subunit vaccines. So that's basically just taking a piece of the bacteria. So not taking the whole cell of the bacteria, but maybe just some of the proteins from the outer coat of the bacteria and putting that into a cocktail and inoculating someone with that. So the proteins are still recognized as foreign substances by the immune system, so you still get an antibody response, but it's only responses against those specific parts of the bacteria. It's not a response against the whole bacteria itself. Okay, so the 2 examples here, acellular pertussis, so that's the kind of pertussis vaccine that's given mostly nowadays. And I have a little story about that, I guess. So when I was a kid, decades ago, the pertussis vaccine that was given was not acellular, it was cellular. So it was a attenuated whole agent vaccine. Okay, and to some small percentage of children that were getting it, they were getting what's called a staring seizure. I don't know if anybody knows what that is, but basically the kids just kind of zone out and just kind of stare up, like at the ceiling or out at the wall, and you can't kind of snap them out of it. And this happens for 10, 15 minutes, and then they kind of snap out of it, and then it can come back. So that was happening to, again, maybe 2 or 3% of the kids that were getting the cellular pertussis vaccine. Okay? I happened to be one of those kids, and it totally freaked my mom out, as you can understand. So when my son was getting vaccinated, he actually got the acellular pertussis vaccine, which is what they're giving now. And the reason they came with an acellular pertussis is that it's people that, or kids that get the acellular pertussis don't get these staring seizures anymore. Okay, so acellular, again, it's just parts of the pertussis bacteria, not the whole bacteria. But what they're finding out, this was within the last 5 or 10 years, they switched to an acellular, is they found out it's not as good of a vaccine. And so we're having a little bit of an outbreak of pertussis now. And I don't know if you guys have seen it, if you have kids, yeah.
Oh, no, I just wanted to ask you, how long does it last, the pertussis vaccine? Because I saw that they're vaccinating adults.
Yeah, so they are vaccinating adults. I don't have a answer for that, exactly how long it's supposed to work. My wife and I were both suggested to get the vaccine when we were having a kid. And when I told the doctor about my episode, he suggested I don't get it. But anyway, what they're finding is that kids that are getting the acellular pertussis aren't as well protected as adults who got the cellular pertussis, back in the 70s and 80s. So what they're finding is that there's an upcreep in pertussis, and you probably see a lot about whooping cough kind of making a comeback, and now they're requiring students to get vaccinated against whooping cough. That's what pertussis is before they can be enrolled in certain public schools. So that's what's happening there. Again, so it's a subunit vaccine. It's not as strong of an immune response because it's just directed against particular subunits of the bacteria rather than the whole organism itself. And the last one, which is a fairly new one and isn't used all that much, is a DNA vaccine. So basically you just inoculate the DNA of the virus itself into the host, and that DNA can either get taken up by the cells, and then the cells use that DNA to make the viral proteins, and then those viral proteins elicit an immune response. Sometimes the DNA itself can elicit an immune response because if it's different enough from human DNA, it's recognized as a foreign substance.
Vaccines may contain different versions of the pathogen. Depending on what they contain, these vaccines may be more or less effective in inducing long-term immunity. Two types of vaccine include the entire pathogen, attenuated and inactivated. In both types, the pathogen has been modified to be less virulent. The attenuated live vaccine contains the living pathogen, but this pathogen has been weakened so that it's unable to cause disease. But the pathogen can still reproduce. This type of vaccine has several advantages. It's the most effective at inducing lifelong immunity. And when the pathogen is a virus, it will induce both humoral and cell-mediated defenses.
So these are two important
The
points here.
disadvantages are that the pathogen can cause disease in an immunocompromised person, and occasionally can revert to its virulent form and cause disease even in a healthy person.
Okay, so I want you guys to get that. So the attenuated vaccine, it's still a living pathogen, but it's been weakened somehow. Okay, so in a normal person with a normal immune system, the bacteria or the virus will replicate a small amount, enough to cause a strong immune response. Okay, this will get the strongest immune response of all the different types of vaccines because you have actively replicating pathogen in your body. Okay, so this one will give you the lifelong immunity. So the MMR is something that you just need a single dose of. And because, like what Sarah brought up, because you actually have replicating virus, if it's a viral vaccine, you actually get cell-mediated defense, not just antibody defense. Because cell-mediated defense requires actively replicating virus. Okay? Now the disadvantages, if someone's immunocompromised, so they have a weakened immune system for some reason, the pathogen, even though it's low dose and weakened, can still cause an active infection if there's no immune system to block it. And there are cases where it could revert to virulent, so if there's some mutation going on, whatever the mutation was to weaken it, if it somehow gets reversed, it could then become a strong virus or bacterial infection again. Okay, so for instance, if they ever come up with an HIV vaccine, they would never use an attenuated live vaccine, okay? Because there's always the potential of getting an active infection.
An example of an attenuated vaccine is the MMR, which protects against measles, mumps, and rubella. The inactivated whole agent vaccine contains killed pathogen. Inactivation makes this type of vaccine safer. There's no danger of causing disease, but the killed pathogen can't reproduce and often has weaker antigens, so it only stimulates humoral immunity and often requires additional booster doses to gain full immunity. The vaccine currently used for influenza is an example of this vaccine.
So I don't know if anybody's ever heard kind of the rumor that some people don't like to get the flu vaccine because they think it actually might give you the flu. It's actually a misnomer. It's the inactivated vaccine, so there's actually no way to get the flu from the flu vaccine. What you can get is if you get a fairly strong immune response. I mean, when you feel sick from anything, it's because your body is eliciting an immune response, and it's causing you to have fever, and all these things are affecting the immune response. So if you get a flu vaccine, you have a strong immune response, you might not feel that well because you're having a strong immune response to the vaccine. You haven't actually contracted the flu. Okay, so I hear that quite a bit. People say, I don't get the flu vaccine because I heard so-and-so got the flu. Okay, it's actually not the case. Now, they have a new thing that, I don't know, a lot of you guys probably heard of, a flu vaccine through a nasal spray. That's actually an attenuated vaccine. So that's actually living flu virus that's given into the nasal passage. And I think that's mostly for younger kids. I don't know if they offer it for adults or not, but I know my son's going to get his flu shot in a couple weeks, and he's going to get the nasal spray.
Toxoid vaccines contain no pathogens, only inactivated toxins from a bacterium. These toxins are treated with chemicals or heat to make them less dangerous, but this also makes the toxin a weaker antigen. Therefore, toxoid vaccines only induce humoral immunity and require periodic booster vaccines to maintain immunity. An example of a toxoid vaccine is tetanus vaccination. Genetic sequencing and recombinant DNA technology have allowed immunologists to delete virulence genes from the pathogen, thus preventing an attenuated pathogen from mutating to a virulent form. Subunit vaccines, such as the one used for hepatitis B, contain only non-pathogenic antigens of the pathogen, not the entire cell or virus. Thus, they cannot cause disease. Conjugated vaccines, such as the one for Haemophilus influenzae B, take the capsule polysaccharides of a bacterial pathogen, which are not very antigenic, and combine them with a highly antigenic protein to gain a more potent immune response. DNA vaccines are currently being developed that contain only the pathogen's nucleic acid. Thus, the pathogen's antigens could be made by the host cells, and they could induce immunity without ever introducing the pathogen itself.
Okay, so you should definitely know the difference between these 5 different types of vaccines, and know the descriptions that go along with them. So here's just a handful of vaccines that are used to prevent bacterial diseases specifically, and what type of vaccine they are. You can see we have some toxoid, we have some subunit ones. Again, given an example, you should be able to determine which one of those 5 types of vaccines there are. Not necessarily the example based on the disease name, but based on the description of the vaccine, you should be able to know which of the 5 subclasses it falls into. Here's some for viral diseases. Again, influenza, it's an activated virus. The nasally administered one is attenuated virus. Measles, mumps, rubella, MMR, we talked about that. The chickenpox is actually an attenuated virus, so it's a living chickenpox virus. Polio is the killed virus. A couple more viral ones, not going to go into each of them. So this is the fairly generic, there's slight changes here and there depending on the doctor. Vaccines that are recommended for children between ages of 0 and 6. So how are vaccines developed? So basically what it requires first is determining the pathogen that caused that disease, and then culturing that pathogen in a laboratory setting, growing it. We've talked about this before. If it were a virus vaccine, you'd have to grow the virus. They're not very easy to grow. You can't just grow them on media like you can with bacteria. Typically they're grown in these eggs. You can see here, all these eggs here with the virus growing in them. That's because they require cells to infect in order to survive. Okay, so there's a lot of controversy and talk about the safety of vaccines. And again, I just want to finish telling you that the one that gives the best immune response is also the one that has the biggest disadvantage if you actually get active infection, if it reverts back to the virulent form. So there's a therapeutic index for each vaccine, and basically that tells you what is the risk of any negative effects versus the benefit of getting the vaccine. And this kind of gives you a way to gauge and compare the different vaccines and determine, you know, which ones are very crucial and which ones can you probably forego because the risk isn't worth the benefit. So it's the comparison of the amount of the therapeutic agent that causes the effect to the amount that causes death in animal studies or toxicity in human studies. So it's again, how much can you give of the vaccine to a person before it causes them to get sick, or if you were doing an animal study before it causes death in an animal study. Okay, so if you talk about, that's the description, if you talk about it quantitatively, it's the ratio given by the lethal or toxic dose divided by the therapeutic dose. Okay, so for instance, can I borrow your pen again?
I think I have some.
Okay, well, you'll just have to listen to me talk then. So the ratio given by a lethal or toxic dose divided by the therapeutic dose. So let's say, if we're talking about a concentration, it takes 100 nanograms of a vaccine to cause disease, but it only takes 1 nanogram to make somebody immune, to give an immune response. So the therapeutic index would be 100, right? 100 divided by 1. Meaning it takes 100 times, it takes 100 times more dose than what you need to be a useful vaccine to actually cause someone to get sick. Okay, does everybody get that? So for a given vaccine, let's say it takes 100 nanograms of vaccine for someone to get sick, or for an animal to die if it were an animal study. And it only requires 1 nanogram in a vaccine to actually elicit an immune response that makes the person immune to that disease. Okay. So the index is just 100 divided by 1. So your therapeutic index is 100 for this vaccine. That would be considered a very safe vaccine. Okay, it takes 100 times more than what you actually need to get somebody sick. Okay, on the other end of the spectrum, let's say again it takes 1 nanogram. You have to give somebody 1 nanogram in order to elicit an immune response. But if you give them 1.5 nanograms, they get sick. Okay, so there's a very small window by what you need to be a useful vaccine versus what it would cause the person to be sick. So in this case, the index is just 1.5 divided by 1. So that would be considered a not very safe vaccine. Okay, because if you just give a little bit more than what's required for the immune response, you can actually get the person sick, or again, it would cause death in an animal study. Okay, so this type of vaccine would be considered very risky, and this wouldn't be the type of vaccine that the general public would get. This would be the type of vaccine that someone in a very specific health condition would be given, okay, where they need to be vaccinated against something for whatever reason, because they have an active infection or there's some other health risk with that person. Okay. Thank you. Okay, so that's pretty much covers the vaccine part of the lecture. Now we'll talk about how immunology, specifically antibodies, are used for research and biotechnology and clinical laboratory stuff. Okay, so diagnostic immunology is basically how we use antibodies in a clinical lab setting to determine if someone has an infection. Okay? So we're actually taking antibodies and using them to our benefit in order to diagnose infection. Okay? So here's an important slide: sensitivity versus specificity. So this is whenever you're developing a diagnostic test in order to determine if someone's ill, you want to make sure it's sensitive. So what sensitive means is that if the person actually has the disease, your test should give you a positive result. Okay? If it only gives you a positive result 50% of the time when the person actually has the disease, it's not very sensitive. Okay, that wouldn't be a good test. Specificity is the opposite. It's a probability that a positive test will not be reactive if the specimen is a true negative. So if someone does not have a disease and they take the test, what is the probability that the test actually says they do have the disease? So in that case, it's not very specific. It's giving you false positives. Okay. So in order to have a very good diagnostic test, you'd want something that has high sensitivity. So anybody that has the disease, it gives a positive response. And it has to have low or high specificity, which means anybody that doesn't have the disease should give you a negative response. Okay. Okay, so the way in a laboratory setting, how we usually make antibodies is we make monoclonal antibodies, which are basically just clones of a single antibody, so they're all the same exact antibody. It's not a mixture of multiple types of antibodies, it's a single type of antibody. And we make them in what's called a hybridoma. So we take a cancerous B cell and fuse it with an antibody-producing normal cell, normal B cell. So we find a B cell that makes the antibody we want, and then we fuse it with a cancerous B cell. Does anybody have any idea why we do that?
Has a very high rate of replication.
Exactly. So basically, it's taking the phenotype of a cancer cell that makes it grow quickly and makes it immortal, so it can continue to divide and divide and divide and divide and divide. And you're fusing that with the B cell that produces the antibody you want. So now you're making a line of B cell that's producing the antibody you want that's immortal and growing quickly. Okay. So how do we do that? We take a lab animal. So let's say we want to make an antibody against an HIV, a protein that's found in HIV. Okay, so we take that protein, that HIV protein, inject it into a mouse. The mouse will give her an immune response against that protein and start making antibodies against it. Usually harvest the spleen, because a lot of the cells making antibodies reside in the spleen. We basically mash up the spleen to make a suspension of spleen cells. The spleen cells, we take those spleen cells and we fuse them with the cancerous cells, and we make this suspension of myeloma cells. And what we get is these hybrid cells. Yeah.
It's not bad that you're using a cancer cell, because aren't cancer cells like damaged?
So you're not using the cells, you're just using the antibodies that the cells make.
The antibodies aren't damaged?
No, the antibodies are not damaged. So you can't just use any cancerous B cell. You have to use a cancerous B cell that's not making antibody of its own. So you basically just have a cancerous B cell that you're using the properties of its ability to be immortal and fusing that with an antibody-producing cell. Okay, so again, now we made these hybridomas, and now these hybridomas, you grow them up in culture, and they'll just be spitting out this antibody that you want. Again, there'll be monoclonal antibodies. Every antibody here will be identical to each other. Okay, so sometimes these monoclonal antibodies can be directly used as medication. So if you guys ever see any type of drug, now not like the advertised name of the drug, like Alexa and all those ones, but the actual chemical name of the drug, anything that ends in MAB is monoclonal antibody, MAB. Okay, so a lot of these are for different types of cancers, for kidney transplant. These are just monoclonal antibodies that are made and injected into patients as actual medication. Okay, so when they're not used as medication, what they're more often used as is for diagnosing disease. Okay, so one of the things we can use them for is what's called a precipitation rate.